Hello AP Biology students, let's go over the 5th section in Unit 6. And in this section, we're going to be talking about the process of transcription. Remember that the content now is focused on the central dogma of molecular biology, which is also the basic flow of information from DNA to a protein. The first step within that process is going to be known as transcription. This is where the cell is going to make a copy of the DNA using a molecule known as mRNA. There are several reasons why DNA itself is not used at the ribosome to produce a protein. One of which is to keep the DNA in the nucleus and not allow it to leave, so in order to keep it safe within that organelle. To begin, we have another enzyme that we have to learn about. This is called RNA polymerase, and it's going to have the job of making a copy of the DNA, again by using mRNA. But RNA polymerase is going to unzip the DNA at a location called the promoter. RNA polymerase will move and make in the same direction as DNA polymerase, and it will make in that 5' to 3' direction. It will use free-floating RNA nucleotides to make this copy of DNA. But as you can see in our diagram, this is RNA polymerase unwinding the DNA, making a copy, and the DNA will actually wind itself back up in this double helix form when it's done. Now, RNA polymerase will make a copy of a specific side of the DNA. The strand that is used by the RNA polymerase is called the template strand. Whenever RNA polymerase attaches to the DNA, it's going to read the template strand. You can see here T's bond with A bond with U, and the C's and G's bond together. So RNA polymerase is going to read the template strand. However, it's going to make a copy of the template strand, which is just going to be like the coding strand. However, it's not going to use thymine. Again, it's going to use uracil. I think of the coding strand as being the actual sequence of nucleotides, since again, the RNA polymerase is going to read the template strand and make a complementary set of nucleotides that's going to be exactly like the coding strand. Again, except the RNA is not going to have thymine, it's going to have uracil. Here's another way of looking at it. You can see again, RNA polymerase, it's going to use the template strand to make a copy of the DNA. Now, this RNA copy is going to be exactly like the coding strand. However, again, it's not going to have thymine, it's going to have uracil. RNA polymerase is going to stop transcribing at a location called the termination site. This again is just another section of DNA where the RNA polymerase is going to unattach and stop transcription. Once RNA polymerase has completed transcription, the mRNA is going to detach from the RNA polymerase. And this is basically where we're going to leave off at, because this mRNA then is going to travel outside the nucleus to a ribosome where it's going to be translated. I think this meme does a great job of showing what connects transcription and translation, and specifically it's the mRNA. Here's another great meme I found in the past year. Protein does all the work, DNA stores all the data, RNA, it's about sending a message. Absolutely love it.